and isn't started.
There we go.
There it goes. Okay, so the meeting is being recorded. Good morning, good afternoon, good evening, depending on where you are. I'm Kevin, and this is my colleague Jan. We're on the video screens right now, and I'm a marketing content manager, and Jan is a senior bioinformatician who will be giving the presentation today on. Let me share my screen. Antibody profiling, utility and interpretation. So we're going to be talking about that today. I'm going to give a short introduction just so you know who we are, our company as well as us individually, and then I will hand the reins off to Jan. A couple of logistics. If you have a question, you can use the Q&A or the chat window. I will ask Jan the questions at the end of the talk. You're all muted, so there's not a lot of chit chat or background noise. Um, so that's usually standard for a webinar, but you can ask questions through chat or Q&A, and please do so. I'd like to make this as interactive as possible. This has been recorded. I know that some people would like to hear what Jan is going to speak about, but are in Australia or California, places where it's very late or very early. So we will make the recording accessible to everybody, and I'll send out an announcement. So if you have colleagues that are interested, this will be recorded. So Jan is one of our senior bioinformaticians. She's a systems immunologist with a bachelor's in science and human physiology and microbiology, and also a PhD that she obtained from the University of Cape Town. She studied infectious diseases, pharmacology, immunology, and she's one of the first people, if not the first people, to map out the proteome in neutrophils. And when did you do that? That was
It was around 2013, 2014. So some other labs had already started doing that, but we obtained the deepest proteome profiling of the human neutrophil at that time.
She's worked with Novartis as part of the Next Generation Scientist program, and she's an alumna of the African Medical Research Council's National Health Scholar Program. She's been working in the field for over 15 years studying infectious diseases and immunology, and her clinical interests center around health optimization, infectious diseases, oncology, and pharmacology. So she has a lot of experience with bench work, with data, as well as with teaching. She's done a lot of lecturing and has run numerous classes. So she will be our guest speaker today. My name's Kevin. My background's in developmental neurobiology. I have a PhD from the University of Connecticut, and I have 13 years of bench experience. I left academics in the mid 2000s and pursued a career as a field application scientist because I liked being in front of people and talking, lecturing, and solving problems. In fact, I'm trying to convince my family to do one of these escape rooms with me. And I think they're up for it. I'm not sure I'm very good at those. I might be the one that gets stuck in the room, but those are the kinds of things that I like to do. I took on the role as a senior marketing content manager with Syngenics about a year and a half ago, and I write white papers and summaries. I'm the one that updates our LinkedIn page, and I may reach out to people to invite them to webinars and tell them about new products and things. What's particularly exciting about the lecture today is that earlier in the month, actually a month ago, we released the IOM Discovery, which is our new protein microarray chip, which has over 1,800 antigens, and it's actually run using IgG and IgA antibody isotypes, so we can identify up to 3,600 or more different data points. And we're intimately familiar with this. In fact, Jan spent considerable time going through all of the different proteins that are on the chip with the bioinformatics team who selected the proteins, and then helping to provide figures like this one to let us know the different types of proteins that are on the chip and the disease areas that are represented. And she knows probably a majority of these very, very well. Protein kinases are highlighted, and those are very important in disease associated with mutations in cancer. They're also very druggable, so there's a good proportion of protein kinases on this particular chip to help you identify those types of things, as well as transcription factors and transmembrane signal receptors, among other things. If you ever have a question about our technology, please feel free to ask. And these different proteins cover a vast range of organ systems: neuronal, dermatological, respiratory, reproductive, and cardio. I'm a big fan of cardio these days because I feel there's a lot that can be done there. And if you have an interest in cardiovascular diseases, please let me know, because I'd like to hear what you're doing and I'd like to learn more about it. My background, like I said, is mainly neuronal, but when I was a postdoc, I started working with gap junctions, and there's actually a connection between gap junctions expressed in the brain and those in the heart. So this was released October 1, and Jan will basically talk a little bit about the background behind this and the antibodies that are involved. If you'd like, I'll put this up later, but this little QR code will link you right to the IOM Discovery page if you want to learn a little bit more about it. So I'll hold this for a second, and then I'll move on with a little bit of background. Okay, so we're going to do a brief overview and then get into Jan's presentation. So one of the things that we excel in and what we really want to accomplish is to try to help bring data to biological meeting. And we focus on functional protein microarrays. This little word cloud based off of one of the white papers that I wrote. So we basically profile antibodies in an individual system using a functional protein microarray. Some people refer to this as immunoprofiling or immunoproteomics. But from this data, we can make lots of interpretations about biological meaning. And what we do as a company is we help you design studies around protein microarrays, the execution of the studies, the statistics involved, analysis, interpretation, and where you might want to go with this data as next steps. So, like I mentioned, our core technology is a functional protein microarray, which has been hugely important in biomarker discovery. And we have among the highest quality arrays on the market with over 15 years. And actually, Jan corrected me, if you combine with many of our other experts, is over 30 years of experience. And what makes these particular functional protein microarrays so valuable is the technology and the thought that was put into manufacturing them. There's a lot of details that need to be considered if you want to get it right. And one of the most important is the CREX protein folding technology that was invented by our chief scientific officer, Jonathan Blackburn, which ensures that the proteins are full length and properly folded, which is going to improve the ability of the antibodies to bind to them, because antibodies recognize shape and charge, and combine those two words into one, and not amino acid sequence per se. So proper folding is imperative if you want to get nice, clean data. So Jonathan Blackburn was one of the founders of Syngenics, and this was a spin out of Cambridge University in 2008. And once the CREX technology was mapped out and they started working on the printing, the manufacturing facility of the proteins was put together in 2016. And we use insect cells to make the proteins. And the slides that we use are actually in an aqueous environment. They're in a gel, so that the proteins literally like float like a kite on them. So that helps make sure that all of the epitopes are accessible and helps maintain the shape and function of the proteins throughout the entire process. In 2020, Summa Equity became a major shareholder in our company. And in 2023, we opened up offices in Boston, and then, I'm sorry, yeah, 2023. And then in 2023 again, as I just mentioned, we started doing the dual isotype standard with the IOM discovery. We've worked with a number of people. Jan knows many of these people very well, including pharmaceutical industry leaders like Glaxo and many academic institutes, such as the National University of Singapore, Weill Cornell, and Weill Cornell in Qatar, NYU, which isn't pictured here, the Michael J. Fox Foundation, among many others. So the CREX technology, as I mentioned, basically it stands for correctly folded recombinantly expressed proteins. And what we do is we encode a biotinylated carrier protein molecule in frame with the protein. So the biotin helps us to adhere the protein to a streptavidin-coated, gel-coated slide surface. And in order for that to happen, the biotin must also be properly folded. If the protein itself misfolds, then the BCC carrier molecule also misfolds, and that masks its binding site. So a misfolded protein won't stick to the slide. So this ensures that all of the proteins are fully functional and fully folded properly onto the slides. And like I said, they're tethered to the slide kind of like a kite so that they are floating in this aqueous environment and all of the epitopes are accessible. This also makes sure that every time we do a printing, the proteins are arranged in the exact same organized manner. It's very consistent, and that's important if you want to get good data. You're not having a protein land on its head or on its ear and masking an epitope. It is always in the same orientation, which makes this a very reproducible type of functional protein microarray. And my last slide is basically, and I'll put this back up at the end, if you want to contact us and learn more, you're welcome to contact me directly. You should have my email from the invitation, but you can also reach out to us through the Syngenix.com website. You can follow us on LinkedIn, and the business development managers are listed here. Chip in the west coast of the U.S., Andy, who manages the entire U.S., mostly East Coast, but he also works with Chip on the West Coast, Adrian for Europe, and Elvira for Europe as well. And then in the Middle East and Asia Pacific, there's Burmet. So I'll put this back up at the end so you have these contacts. And with that, I'm going to hand it over to Jan to continue the presentation.
Okay. Hi, everybody. Thanks for joining today. Let me just take a moment to share my screen. Okay. Kevin, can you see the presentation, but not the little mini window, the little mini Teams window?
Yeah, I see your main window, the presentation window.
Only the presentation. Okay, perfect. Thank you. Okay, so I thought that this might be a good topic for today. These are the types of questions that we get a lot from our collaborators and from potential partners that want to work with us. What is the utility of antibody data? How can you use it? How is it relevant? What are the applications? And then also to think a little bit about, once you have the data in hand, what does it mean? How can you get from the raw data and then the results through to biological meaning and clinical utility? So this is going to be a pretty short presentation. I think it's about 10 or 12 slides. And the general structure is we'll talk a bit about the general utility of antibody data. Then we'll go through just a few applied examples, both from our platform and from other platforms. And then we'll talk a little bit about how to think about antibody data to be able to get from data to meaning. And then I believe there'll be a few minutes for Q&A at the end. Okay, so we'll jump right in. How are antibodies relevant to your research, potentially? So before I get into the actual slide, I just want to say, really similarly to the immune system and how it is relevant to almost every clinical sphere, if not every clinical sphere, antibodies are analogous. Almost any condition you can think of, they have some kind of relevance. And so it's very difficult to truly give a comprehensive overview of all of the utilities of antibodies and antibody data. But what I've tried to do here is just kind of categorize them into bite-sized buckets. And we'll just talk through those categories. And obviously there are additional, more specific applications. And we'll just briefly touch on some sort of blue sky thinking applications at the end of the slide. Okay, so one of the things that you can do by profiling antibodies in a sample is you can identify targetable antigens. And this could be useful, for example, if you're trying to develop an infectious vaccine or a cancer vaccine, and also for various other purposes, for example, for CAR T cells, CAAR T cells. You can also, by screening the antibodies in your sample, you can identify potentially targetable pathogenic antibodies, as well as potentially exploitable protective or therapeutic antibodies. Something that I'm particularly interested in, you can identify antibody data-driven molecular subtypes, which then hopefully associate with sociodemographic or clinical or laboratory phenotypes of interest. And we do see this happening frequently. I'll show an example of that in a couple of slides' time. And then, of course, we can identify clinically relevant antibody biomarkers of any type. So these can be predictive biomarkers, diagnostic biomarkers, or monitoring or surrogate endpoint biomarkers. We find that the interest at the moment seems to be mostly in predicting treatment response. So for personalized medicine approaches and also for patient stratification for clinical trials, and then also predicting immune-related adverse events. Then if you switch tack a little bit from the more clinical perspective and you put on your sort of academic hat a little bit more, and you're really trying to understand a complex dynamic and sometimes immune-driven disorder, you're trying to understand the mechanisms, you're trying to understand the etiology and the pathogenesis, then the applications fall more into this functional bucket that's in the middle of the slide. So antibody data can help you to start inferring hypotheses regarding your underlying disease etiology. And most specifically, the first question you can ask is which of the finite number of antibody elicitation mechanisms were likely involved in your particular case. It can also help us to start inferring hypotheses regarding the underlying mechanisms of either protection or pathogenesis or progression of a state of health or disease. And then, as I mentioned, there are many, many more custom application examples and specific applications. And here, one can really be extremely creative, scientifically creative in thinking about how to apply this technology and how to apply the antibody data. So you could think, for example, about using protein variants or protein binding partners to demonstrate the phenomenon of epitope and antigen spreading, for example, following some kind of therapy like immunotherapy. You could think about exploiting B-cell receptor or antibody specificities that have been identified using this kind of technology to help you to identify tumor-specific T cells or other specificities of T cells in other disorders. I do have more in this vein of custom examples, but I think let's move on and we can always talk more about that in the Q&A if anyone's specifically interested in that. All right, so which disease areas is this technology applicable in? Which disease areas are relevant to the antibody world? Pretty much all of them. I've listed the ones here that we most commonly get questions about, that we most commonly work with. So infectious diseases, obviously. Also microbiome research, maybe slightly less obviously, and I'll touch on that again in the next slide. Rheumatology and dermatology. Oncology, including paraneoplastic syndromes, neurology and psychiatry, cardio and cerebrovascular disease, endocrinology, allergology and immunodeficiency. And then the next two categories, therapeutics, drug targeting, and PPI, protein-protein interaction. That's not necessarily something where you're going to take a biological sample and profile the antibodies in it, but you may wish to look at the binding partners of your small molecules and your biologics. You may wish to look at the binding kinetics. You may wish to look at off-target binding of these small molecules or biologics or other proteins that you are interested in finding out which other proteins they interact with. So that's a slightly different category of application, but no less applicable. And then as well, genetic disorders, including rare genetic disorders and general difficult to diagnose disorders as well. This technology and also antibody data in general are very applicable there. You can see I've just put a number of examples here of recent publications that sort of touch on each of these areas, just so that you can get a feel for what is happening in the field. The very first publication that you see up at the top in the middle, that was done on our platform, but all of these other publications, some of them are review articles, all of the other primary publications were done on a variety of different platforms, ELISA, immunofluorescence, other types of custom protein microarrays. So it's not all focused just on protein microarray technology. I think I just want to read one or two of these headlines just so that you can get a feel for some of the interesting things that are happening in the field. So this one in particular, population-wide diversity and stability of serum antibody epitope repertoires against microbiota. So this is in the top right-hand corner of the screen. And the reason I wanted to touch on that one is the stability aspect, because something that perhaps I'm preaching to the choir, but something that perhaps the audience doesn't know is that a large proportion of the antibody repertoire is actually quite stable over time. And so if you measure my autoantibody profile now and you measure it in 5 years' time, even 10 years' time, a large proportion of that profile will still be the same. And it will be sufficiently the same that if we put the data in a group of data of other people's antibody profiles, my two samples will cluster together with each other and your two samples will cluster together with each other. So there really is incredible stability of the autoantibody profile and the general antibody profile over time. And it's really about 2 to 8% of that profile that is actually perturbed by any specific disorder, disease, therapeutic intervention, lifestyle intervention. And that is the percentage, that small proportion that changes, that is the percentage that we're interested in looking at from a disease perspective. Okay, let's go on to the precision medicine aspect. So I think it bears saying this explicitly because it's, you have to make a couple of mental leaps to actually make these connections. So I think it's helpful to think about it in this way. If you think about medicine traditionally, a lot of our clinical laboratory tests are, for example, based on reference ranges. So you could think about reference ranges for albumin, for example, or reference ranges for minerals, or even for vitamins. And those reference ranges were established by looking at levels across healthy populations. And that has been extremely helpful. It's been a really good springboard for a lot of medicine. But in certain areas of physiology and biochemistry, it's not really possible to establish those kinds of reference ranges. So I'm thinking about certain aspects of immunity and also the microbiome, where there is just this incredible inter-individual heterogeneity, which is probably related to genetics, ethnicity, HLA profiles, lifestyle, and so forth. But that makes it very, very difficult if you have established someone's microbiome profile. For example, how do you know what is normal, what is good, and what is bad? How do you categorize the information that way? And so we really haven't had a way to do that. But one of the ways to look at this is to think each individual's immune system is constantly surveilling both inside cells and outside cells. And the immune system, to a certain degree, knows what's normal for each individual, and it will report on what it thinks is abnormal for that individual. And so if you combine that kind of information with, say, microbiome information, you can start getting a sense for what is good and what is bad, or what is normal and what is abnormal for that individual. In addition, instead of using, a multi-omic technology, a true multi-omic technology like proteomics, where you're really aiming to profile all of the proteins in a sample and all of the changes in proteins associated with a particular disease, in which case you are going to be looking at both drivers of disease and passengers of disease that are not actually related to the disease. So instead of doing something like that and then really having to sift through the data and trying to categorize, which are drivers and which are passengers, immunoproteomics starts helping you to home in on the drivers of disease because of this ability of the immune system to pass the information, the in vivo information for what is normal and what is abnormal. And if you think specifically about the antibody component of the immunoproteome, it can actually directly identify immune targets for you. So things that the immune system is targeting. Those targets are not incidental. For the most part, those targets are not incidental. They are the result of a process, a causal molecular process that has resulted in elicitation of an antibody of that specificity. So we like to think of our technology, but also really any multiplexed antibody profiling technology as a good first screening tool that can then inform which next step you want to take. Do you then want to go and do some kind of proteomics, whether it is a shotgun type of proteomics or a targeted type of proteomics? Do you then want to follow up with immunohistochemistry? And in saying that, I also just want to make clear that multiplexed autoantibody profiling or general antibody profiling is very complementary to all of these other tools. So it's not a replacement. It is something that works in tandem with these other tools. And I think the very last slide of my presentation will also briefly touch on that as well. So if you look at all of these disease-associated changes that one might find, altered antigen sequences. So for example, in the cancer field, you might find something like a mutation, or, in the field of just heterogeneity in health, you might find sequence variations. So altered sequences, antigen over or under expression, aberrant splicing, aberrant proteolysis, inflammation, all of these disease-associated profiles. We like to think of antibody profiling as being able to screen for all of these, and then you can go and look deeper into whatever it is that whatever the data suggests you should look more deeply into. Okay. Just in case anybody in the audience isn't familiar with protein antigen microarrays, I just thought I would give a quick conceptual overview of how they work. This isn't unique to our platform. There are many other smaller custom arrays that you'll see in the literature that people have developed for individual experiments. But I just want to give an idea of the steps of the assay and how it actually works. So you'll see at the top of the screen is a glass slide. Bound to the glass slide is a native protein antigen. The surface is non-denaturing, so the protein stays in its functional native conformation. Then the second item below that is a Y for an antibody. So that represents an antibody in your biological sample that is then going to bind to its cognate protein antigen. Then the next thing down is a Y with a little star, and that represents a secondary fluorophore labeled antibody, which then binds to the antibody bound to the antigen to be able to give us a signal. And so the result that you get from this type of protein antigen microarray experiment is you learn about the antibody specificities in your sample, and you also learn about the relative quantities of each of those antibody specificities. Okay, so I've given you an overview now of how antibody data can be useful. So I just want to talk you through a very few examples, applied examples. So these particular two were done on our platform. I will also take you through two that were done in a completely different way, just so that you can see that antibody data is useful across the board. So here we have the first study by Patel et al. From 2022. This was done at the University of Birmingham, or in collaboration with the University of Birmingham rather. And what they did was they identified an autoantibody signature predicting post-resection survival in non-small cell lung cancer. So they had a really nice big discovery cohort, 111 participants, both survivors and non-survivors. And they had a nice validation cohort as well, also survivors and non-survivors. And they were able to identify, using the Syngenix CREX technology, a 13-marker autoantibody panel that significantly and independently predicts survival with a sensitivity of above 80% and a specificity of above 70%. Then if you look at the study on the right-hand side, so this was Miles Lewis et al. This was from 2018. This is one of my favorite studies because it touches on molecular subtyping, and I'll talk a bit more in detail about that in a moment. But this was done in collaboration with Queen Mary University. What they did was they identified what appeared to be clinically relevant molecular subtypes of lupus. Again, they had a really nice large discovery cohort, including matched controls, a nice large validation cohort, including matched controls. And what they did was they identified 79 lupus discriminating autoantibodies, 11 of which were known and 68 of which were novel. And they also found 4 clinically and mechanistically relevant subtypes using this antibody data. So I'm just going to tell you a little bit more about that study now, specifically about the subtypes and how they were clinically and mechanistically relevant. Okay, so these are figures taken directly from the publication, and these are the 4 subtypes that were identified. So if you look at the heat map on the top right-hand side, you can see that the annotation bar across the top of the heat map shows you the titles of each subtype, so SLE3, SLE1B, SLE2, and SLE1A. And here on the left-hand side of the slide, we have some data about each of these subtypes. So subtype 1A, which is red on the heat map, is associated with pulmonary symptoms. This other little heat map E, this is where you can see the associations between the subtypes and the clinical symptom clusters. But anyway, so 1A, subtype 1A is associated with pulmonary symptoms. The subtype seems to be driven on the heat map by high levels of anti-SSB and anti-TROV2 antibodies. And then the targets of the autoantibodies in this subtype are enriched for RNA processing and RNA dynamics functionalities. And this is the dendrogram. So the image number C on the bottom right-hand side of the slide. The dendrogram tells us more about all of the different targets and each of the functional groups that are enriched. And you can see at the bottom of that dendrogram that all of the targets in red correspond to subtype 1A that we've just been discussing. Subtype 1B, this subtype, so this is in purple on the heat map at the top. This is associated with renal symptoms. The antibodies or the antibody signature that drives this subtype is high levels of anti-PSMA3 and anti-RQCD1. And the targets of the antibodies that are enriched in this subtype are also enriched for the functionality of RNA processing and dynamics again. Then subtype 2 is associated with arthritis. Multiple antibodies drive this subtype. It's not as clear-cut as the other subtypes. And the targets of these antibodies are enriched for TGF-beta and Wnt signaling pathway members. And then finally, subtype 3, also defined by multiple antibodies. This subtype didn't have a specific clinical symptomatology association, but the targets of the antibodies were enriched for TLR2 NF-kappa B and apoptotic signaling, as well as for lymphocyte development functionality. And so, you know, this starts getting at the power of this kind of data. And, you know, this is not the only data set that we've seen like this. So maybe let me say two separate things here. One thing is that these subtypes that are SSB, TROV2, PSMA3, RQCD1, etc. driven, we also see those kinds of subtypes in other published studies. So this is not unique to this particular paper. In some ways, it has been validated, although not with regards to association with symptoms, but just the fact that lupus does have these specific antibodies that are driven by these particular autoantibodies. And the other thing is that we see frequently across lots of different types of disorders, when we apply a subtyping analytical strategy to the data, we see that there is this underlying heterogeneity. So even though the clinical nosology has said, okay, everything that has these symptoms looks like this disease, and we're going to group it together as lupus, or as rheumatoid arthritis, or as a particular type of cancer, or whatever it is, underneath the hood, there actually genuinely do seem to be molecularly different subtypes, distinct subtypes. And whether or not those subtypes are due to etiological differences or due to a different time point in the continuum of disease development at the time that you measure that patient, or due to lifestyle factors or due to treatment factors, that is less clear. But I hope that over the next few years, we will really start elucidating the reasons for this underlying heterogeneity in these complex disorders. Okay, these are the last two sort of applied examples that I want to show. So I've specifically selected immune-related adverse event prediction and treatment response prediction, because as I mentioned, this seems to be where most of the interest is currently. So neither of these two studies were done on our platform. The study on the left-hand side is a retrospective study that was done at the University of Chicago Medical Center. And what they did was they identified autoantibodies that predict immune checkpoint inhibition therapy induced adverse events in cancer. So of the very large cohort that they identified in their records, 135 of them had no previous autoimmunity, and they had one or more rheumatoid antibody test results available. What the study showed was that if a patient had a pre-treatment rheumatoid factor positive test, That's significantly associated with post-treatment rheumatic-related adverse events, with an OR of 25, which is pretty high, a confidence interval that doesn't cross 1, and a p-value that is significant. In addition, they said as their conclusions that both pre and post-treatment rheumatoid factor were 93% and 78% specific for immune checkpoint inhibitor-induced arthritis. And this was a study from very recent 2023. Then the study on the right-hand side, Shen et al. 2023 as well. This was done at the MD Anderson Cancer Center. This is a very interesting approach. They identified potential content for a custom chip, so a custom antigen microarray, and the content consisted of frame shift peptides that were predicted based on the dysregulation of mRNA processing in cancer. And so they added, approximately 375,000 tumor-associated frame shift peptides to their array, and they assayed patient samples, lung cancer patient samples prior to immune checkpoint inhibitor therapy against this array, looking for seroreactivity to these frame shift peptides. And the anti-frame shift peptide antibody signature that they identified ended up providing 98% accurate prediction of disease progression after treatment. So there really does seem to be some very strong, somewhat latent value in the autoantibody repertoire that one can get at through many different technologies and using many different approaches. I just want to switch to the next. Okay. I just want to finish off the sort of applied example section of the presentation by saying that there clearly is added value of isotypes and subclasses rather than just looking at antibodies broadly or at a particular single isotype. In that, if you look at specific isotypes, and especially more than one, and specific subclasses, and especially more than one, you can really start improving your disease resolution in terms of your clinical sensitivity and specificity. It's relatively easy to measure multiple isotypes or subclasses simultaneously. You often find that you're maximizing the utility of your data because the different isotypes and subclasses don't target the same targets. There is some target overlap, but there is often quite a lot of unique targeting by different isotypes and subclasses, which makes sense given the potential differences in antibody elicitation for different isotypes and subclasses. If you measure multiple isotypes simultaneously, it can give you temporal clues as to where in the continuum you are. Is this potentially an acute response, or is it a response that is still developing, or is it a memory response? And it can also give you compartmental clues. So if you think, for example, about IgA, what are the reasons that you might have a systemic IgA response? Possibly the likeliest explanation is that this response was elicited at the mucosal interface or in the skin, where there may have been some kind of breach of those barriers. Okay, so I'm not sure exactly what the time is. Kevin, can you give me a quick heads up as to how much time we have left?
It's 9:40, so I would say 7 minutes or so.
Okay, perfect. So I'm gonna try and just blast through the getting from results to meaning in those 7 minutes. Okay, so obviously, in order to get good results from an experiment, there are a number of steps you have to follow. Some of these are common to almost any research, so it's not necessarily specific to antibody research, but it bears repeating nonetheless. So we like to start with impeccable experimental design that is commensurate with the research question. If you don't design your experiment appropriately and you try and ask all kinds of ad hoc questions from the data later, you're going to run into problems, for example, with statistical power, for example, with what you're actually able to derive from the data. Once you've run your experiment, you've designed your experiment appropriately, you've used the appropriate sample types, you've run your experiment appropriately, then you also need to handle your data appropriately. And we also help our customers and our partners do this, or guide them to do this themselves. And as I mentioned earlier, the output data that you expect from an antibody profiling experiment is the target specificities of the antibodies and also the relative quantitation values. So once you have those data in hand, the question then becomes, how do you interpret them? What can they tell you? How do you think about them to get from the data to the meaning? So the first thing you might want to do is look at your results and filter them for the most experimentally or statistically convincing results, as well as, if you are doing a biomarker study, for ideal biomarker characteristics. Then you can maybe tabulate the results and you can start making biological inferences in the form of testable hypotheses, specifically regarding the antibody elicitation mechanisms and what their in vivo functions might be. I'll talk about this more on the next slide. And the way to think about the directionality of change, if antibodies are elevated in cases or if they are depressed in cases, there are different ways to think about this depending on the disease context. So in the context of an infectious disease, for example, if you have an elevated antibody to a particular pathogen, then that suggests that the pathogen is eliciting that antibody, and that's why the antibody is elevated. However, if you have something like long COVID or something else where immune complexes are implicated in the disease process, and you have an elevation in the cases, it doesn't necessarily follow that the elevated antibody levels are because the antibodies are currently being elicited. It might be that your comparator class, and I'm saying here in controls, but the comparator class could be something like a different grade of disease. So in those cases, it might be that the antibodies in your comparator class are actually being consumed into immune complexes and therefore are less available for assay. And so it just bears careful thinking about the directionalities that one sees relevant to the context of the disorder that one is studying. Once you have those testable hypotheses, you can start doing literature searches. Has anyone else found anything similar? You can maybe do some kind of empirical verification. If you want to do an orthogonal method, make sure that the antibodies you've identified by a microarray experiment are also being picked up in an ELISA, for example. And if it's a biomarker discovery experiment, perhaps you want to then move on to a validation experiment and further clinical validation studies. Okay, so briefly, let's talk about antibody elicitation mechanisms. If you look at the antibody image almost in the center of the slide, it is surrounded by all of the currently known or suspected antibody elicitation mechanisms. There are some antibodies that are germline encoded and that are constitutively produced. They don't need to be elicited by a stimulus. They could be elicited by some kind of sequence variation or structural variation, altered level, ectopic presence, aberrant post-translational modifications, aberrant proteolysis, aberrant folding or aggregation, and molecular mimicry. It's useful to think more deeply about antibody elicitation, not only in terms of the etiology of a particular disorder, but also in terms of how one would approach therapeutics in, say, autoimmunity. So if you just look at the little box on the left-hand top side of the screen, this is just a reminder that B cells, naive B cells, can respond to all manner of molecules. It doesn't have to be a protein antigen. It can be a carbohydrate antigen or a lipid antigen or a nucleic antigen or a combination. These molecules can be soluble, or they can be presented by antigen-presenting cells, or they can be in some other way associated with the cell surface. And they can be part of molecular complexes or aggregates of abnormal aggregates of proteins. And all of these responses can occur either with or without T cell help, depending on the category of antigen. And so the reason I'm restating this is that there seems to be a slow shift happening in the thinking around what constitutes a B cell targeted autoantigen and how that type of B cell mediated autoimmunity comes about. Classically, it really was thought that central tolerance mechanisms were sort of the main mechanisms driving tolerance, and that peripheral tolerance mechanisms were just kind of a backup. But I think that picture is slowly shifting. So as we gather more and more evidence about the molecular characteristics of antigens, and as we understand that it doesn't seem to be the entire human proteome, but rather just a subset of the proteome that seems to become an autoantigen or seems to become autoantigens, it becomes clearer that molecular mimicry and also local inflammation-induced changes and sequence or structure-specific changes may actually be the major drivers of the mediated autoimmunity. And so that would suggest that although central tolerance is obviously helping to establish tolerance, that it is by no means the case that all proteins are, all host proteins are tolerated because of central tolerance. There are, in fact, proteins that are more prone to becoming autoantigens, and this has to do with their structural characteristics, as well as whether or not they may be present in a context of local chronic inflammation. So if you look at the box on the lower right-hand side of the slide, you can see a number of molecular characteristics that are associated with known antigens. And these are things, and this is very telling, these are things like being extremely highly evolutionarily conserved, repeated molecular patterns, especially on the surface of the antigen, the fact that these proteins are often nucleic acid binding proteins or other protein binding proteins, or rather just proteins that bind things. And also many of these proteins are immune pathway associated proteins. So if you're anything like me, if you see those kinds of characteristics, your brain already starts to try and make causal leaps to say what is underlying those characteristics. And I'm not going to lay it all out explicitly for you, but you can think about it, and it starts becoming quite obvious. The funnel at the top here is just showing that although we have so many proteoforms and so many protein-encoded genes, we only know about approximately 800 confident autoantigens that have appeared in the literature at least twice. And obviously this number is likely to increase somewhat, but it really does seem that there is a subset of the proteome that's prone to becoming autoantigens. Okay, very briefly, not going to go through the slide in great detail. Then you can also start thinking about what are the most likely in vivo functions of any antibodies that you've identified in your study. Down the side here in green, we have all of the typical antibody functions that you expect that are helpful, for example, in an infectious disease context. On the right-hand side in red, you have all of the dysfunctional mechanisms that we know of that antibodies can engage in, sort of the classical mechanisms. And then we also have some mechanisms that can belong to either of these. So uptake of certain immune complexes can modulate cell fate and function in different directions, both in protective and in pathogenic directions. There are additional antibody functionalities. So for example, here in 2020, there was a study that showed that direct interaction of an antibody with a TNF receptor induced programmed cell death. And there are additional functions. Some antibodies are directly proteolytic, for example, which is surprising if you sort of think you know about antibodies and you come across that. Finally, if you think about antibodies elicited in vivo in your condition of interest, there are a number of categories that they might fall into. They might be protective in your state of health or state of disease that you're interested in. They might be primary pathogenic antibodies, i.e. they are responsible for the disease. It is those antibodies that are the etiology or that are responsible for the etiology of the disease. Or there might be secondary pathogenic antibodies, which is that they are elicited by the disease process. They're not responsible for starting it off, but they are elicited and then they become active in the pathogenesis as well. Finally, they can be incidental, so they can be elicited by the disease process. They're not responsible for starting it off. They're only elicited by it, i.e. they report on the disease process, but they don't become incorporated into the pathogenesis, but they're still useful as reporters. I do have examples here of each of those types of antibody or disorders in which, or treatments in which each of those types of antibodies are relevant, and their likely subcellular locations. Okay, the very last slide, which won't take long, is I just wanted to hammer home the point that antibody data is extremely valuable and can be extremely useful, but it can be even more useful if you combine it with other omics data or other antibody functional determinant data. So for example, isotype subclasses, the glycosylation state of your antibody, the subcellular location of your antibody, and so forth. And you can imagine, for example, a situation in which you have a proteomic data set that shows you that a certain protein is abnormally upregulated. Perhaps you also have a data set that shows immunohistochemistry data set that shows you where that particular protein is upregulated, which biological compartment. And you have an antibody data set that shows you that you have antibodies that are of the right inflammatory kind of subclass and isotype, and they're targeting that antibody, and they have characteristics of autoantigens, or they have characteristics that make them more likely to precipitate and cause disease. Together, those data sets then are quite a strong indicator that that is probably how the pathogenesis is occurring. Whereas any of those data sets on their own, it's more difficult to understand. So I just wanted to say that orthogonal data integration can really help to extract maximal utility from samples that are available. And this is also a particular interest for the bioinformatics team at Syngenics. So if this is something that you're interested in and you want to talk more about orthogonal data integration, particularly of multi-omics data sets, then we're also happy to chat about that from just a blue sky science perspective. Okay, so that is my whole presentation. And I think we have maybe a few minutes left for questions. Kevin, can you guys, okay, you are still
Yeah,
there.
I can hear. No, I can hear. I was just reading the comic. Thanks, Jan. That was fantastic. I really appreciate the depth of knowledge that you shared with everybody today. One question that came in, and please feel free to ask questions, is will the slide deck be shared? Are you able to share this particular deck? Okay, so we can.
Yeah, absolutely. I just need to clear out my personal notes in the boxes below the slides, but I'll do that and I'll share it across with you, Kevin, and then you can make it available.
Okay, thanks. I was kind of curious about some things as I've been learning more and more about this over the last year, and that is the relationship between the autoantibody profile and the functions of the proteins. I'm not sure if you could go into too much detail, but one thing that I was hoping when I learned this type of information is if we segregate a population of antigens that are indicative of a disease, can we then target those proteins for potential therapeutic? And how would you go about that? How would you think about, say, with the Patel paper, there are 13 autoantibodies. Where would you start if you wanted to develop therapeutics?
Yeah. So in order for something to be druggable or targeted as a therapeutic target, it has to in some way be involved in the pathogenesis of the disease. So let's say we identify some autoantibodies that seem to very clearly define a particular disease or disease subtype. And we think about how those antibodies may have been elicited. And we can tell that the protein targets of those antibodies, like we have an orthogonal data set, for example, that shows that the targets of those antibodies are either upregulated in an abnormal way or being expressed in an abnormal compartment or are being aberrantly, they have an aberrant sequence, so they're being aberrantly modified or something like that. So the point I'm trying to make is that you do need additional data in addition to the antibody data to sort of confirm your hypothesis that the reason the antibodies are targeting those pathway members is because the pathway members themselves were dysregulated in some way to start with, and that's how the antibody was elicited. If you confirm all of those things sufficiently, then you can start thinking about that protein target, the antigenic target, and the pathway member as potentially a therapeutic target. I know this is the case in a lot of TNF-driven disorders. We often see autoantibodies to the TNF signaling pathway members, lots of different members of the TNF signaling pathway, not just one. And many of those pathway members are in fact therapeutic targets, which are currently being targeted by drugs that are on the market. And some of them will be targeted by new drugs or by old drugs that are being modified, well, not modified, that are being relicensed for new indications or reinvestigated for new indications. So it's definitely a process, but absolutely the antibody data can be your first indication that a particular antigenic target might be something that could be therapeutically targetable, or that the pathway it belongs to might be pathogenic in that disorder and therefore might be a therapeutic target.
Thank you. When I went to share my screen, the Q&A disappeared, and that's it. So I will reshare
No
this
more
screen.
questions? Please
No
feel
more,
free.
no more questions at this time. But if folks would like to write in a question, maybe you think of something tomorrow over coffee, please feel free to email me or note me through Teams if you can do that, and I will pass it along to Jen. This is, again, the list of contacts on the screen. We're building a webinar series, so stay tuned for future webinars. Wanted to talk about the history of antibodies in one, and also neuro and aging are emerging, and I think are starting to appreciate the role of autoantibodies in those particular disorders. So stay tuned, and everybody, thank you for joining us, and have a wonderful day.
Thanks so much for everyone's time.
Bye.
Bye, everyone.